There are two separate populations of ribosomes in the cytosol. Free ribosomes, which are free in the cytosol, and membrane-bound ribosomes, which are attached to the cytosolic side of the endoplasmic reticulum membrane. For our purposes, this ribosome was a free ribosome with a signal sequence which directed it to the rough ER to become a membrane-bound ribosome. The mRNA carries the transcribed DNA to the ribosome, and the ribosome and the mRNA come together to begin translation. Two protein components help guide the ER signal sequence to the ER membrane. A signal recognition particle present in the cytosol binds to both the ribosome and the ER signal sequence when it emerges from the ribosome. Then, the second component, the SRP receptor embedded in the ER membrane, recognizes the SRP. This binding of an SRP to a ribosome that displays an ER signal sequence like ours slows protein synthesis by that ribosome until the SRP engages with an SRP receptor on our ER. Once those two are bound, the SRP is released, and the receptor passes the ribosome to a protein translocator in the ER membrane, and protein synthesis begins. The polypeptide is then threaded across the ER membrane through a translocation channel. In addition to directing the proteins to the ER, the signal sequence functions to open the channel in the protein translocator. It replaces the translocation channel plug and remains bound to the channel, while the rest of the polypeptide chain is threaded through the membrane as a large loop. Most proteins are covalently modified in the ER. One of these modifications is glycosylation. This occurs when a 14 preformed sugar oligosaccharide consistent of glucose, mannose, and N-acetylglucosamine is added by oligosaccharyl transferase from dolichol phosphate to asparagine on the growing polypeptide chain. The ER signal sequence is removed by a transmembrane signal peptidase, which has an active site facing the luminal side of the ER membrane. The cleaved signal sequence is then released from the translocation channel into the lipid bilayer and rapidly degraded. Once the C-terminus of the soluble protein is passed through the translocation channel, the protein will be released into the ER lumen. Also, as a peptide is synthesized, it begins the process of folding and quality checks in the rough ER. First, glucose is trimmed to enable interaction with calnexin, which is a molecular chaperone. The calnexin binds to the terminal glucose residue of a protein to allow for correct folding or refolding, and protein disulfide isomerase catalyzes the formation of disulfide bonds. A glucosidase removes the terminal glucose, freeing the protein from calnexin, and glucosyl transferase checks for proper folding. If it is folded properly, the glucosyl transferase adds the terminal glucose and restarts the folding process. If it is properly folded, one mannose is removed, and the protein can now exit the ER and head to the Golgi apparatus. Transport vesicles move folded proteins from the rough ER to the cis Golgi and then through the Golgi, where further modifications occur, and to the trans Golgi, and then to their cellular destination. A vesicle will bud from the Golgi and will weave through a process of exocytosis, and then they travel to their cellular destination through a four-step process. First, the vesicle moves toward the target location along the cytoskeleton. Then, Rab and tethering proteins are used to attach the vesicle to the target membrane. Docking occurs by T snares, which are attached to the membrane of the cell, and V snares, which are attached to the vesicle. And lastly, fusion occurs between the vesicle and the target membranes because the T snare and V snare twist together with significant force, which causes the two membranes to fuse together.